Transcription, as I told you, it's the process of formation of RNA by using DNA, as a template. Transcription copies DNA to make a strand of RNA. Strand. What the name of the enzymes in transcription? The name of the enzyme is RNA polymerase. RNA polymerase. If you remember the name of the enzyme in replication, it was DNA polymerase. But here in transcription, RNA polymerase. What's the role of RNA polymerase? RNA polymerase, with the help of other proteins, they form something called transcription complex. The complex, the union that has the function of performing transcription process. The transcription complex recognizes the start point in the DNA to unwind or unzipping the DNA, but unzipping a certain gene, a certain part of the DNA, the part that carries the gene that is responsible for a certain protein the cell needs. Look here. Here it is. This is the DNA. But now the cell wants only this gene. So transcription occurs for only a certain segment, for a certain gene that carries information for a certain protein the cell needs. Here is the complex, the RNA polymerase and other proteins. They will start to bind to the start point in the DNA to unzip the DNA or unwind the DNA. So the first step here, like in replication, unzipping or unwinding, but here for the segment, not for the two strands, not for all the DNA. After that, the complex, the transcription complex, will start to get nucleotides or to help the nucleotides to pair with one strand of the DNA. Now, for example, this is the strand that will be used by the transcription complex to form the new strand of RNA. This is the red one, is the RNA. The transcription complex will start to bond the nucleotides together, but bond the nucleotides on the RNA strand. Here it is the RNA strand. They will bind nucleotides in the RNA strand, but there are no hydrogen bondings to join the new strand with the old strand like in replication. No, here only bonds to bond the nucleotides on the same strand. After that, the DNA helix, after finishing, for example, transcription finished in this area, again winding or again zipping of the DNA to go back to its original state. And after that, here is the RNA strand is ready. The RNA strand now is ready. So the RNA strand detaches from the DNA once the gene is transcribed. Here is the RNA for this piece only, because this piece only for a specific gene. After that, the RNA will detach and the DNA will wind again, or zipping of the DNA will take place. This is the process of transcription. The process of transcription takes place in the nucleus, inside the nucleus, where the DNA is stored inside the nucleus. After formation of the RNA will start to leave the nucleus to start the process of translation, which is the formation of protein from RNA. So the process of transcription takes place in the nucleus. The RNA has three types, not only one type. The first type is called messenger RNA, mRNA. mRNA carries the messages that will be translated to form a protein. So mRNA, M for messenger, it carries messages in the form of codes or codons. Ribosomal RNA, here is the messenger RNA. This is the messenger RNA. Single strand of RNA carries codes. Those blocks are codes for the formation of a certain protein. After that, ribosomal RNA. From its name, ribosomal, so rRNA forms part of the ribosome. It forms part of the ribosome. This is ribosomal RNA or rRNA. So now we finished two types, mRNA, rRNA. Let's see tRNA. tRNA, T from transfer RNA, tRNA, transfer T. So what do you think the function of tRNA? It's like a truck to carry amino acids. What are amino acids? Amino acids are the monomers for the formation of proteins. So many amino acids together, amino acid linked to another amino acid will form the protein. So tRNA is the truck or the transfer RNA that brings amino acids from the cytoplasm to a ribosome. Later on in the coming lesson, inshallah, we will talk about tRNA. So tRNA brings amino acids from the cytoplasm to a ribosome. tRNA transfer, you can remind yourself that this is tRNA, like a truck. Here it is. This is the tRNA, like a truck. It carries, this is the amino acid. Like a truck carries the amino acid to go to the ribosome to form the new protein. Now let's talk again about the similarities and differences between replication and transcription. Number one, transcription and replication both involve complex enzymes. Transcription, RNA polymerase, replication, DNA polymerase, and in both cases there are complementary base pairing. The two processes have different end results. Replication end results, the DNA, formation of copies of DNA. What about transcription? Formation of RNA. So they are different in the end results. Replication copies all the DNA, not only a piece of DNA, no, copying of all the DNA before division. While transcription copies a certain gene when the cell needs a certain protein, so transcription will occur for a certain gene in the DNA, not for all the DNA. Replication makes one copy, so after replication, one double helix will form another double helix, while transcription can make many copies according to the cell needs. So the number of copies of RNA depends on the cell needs for the protein. Those are the differences and similarities between replication and transcription, and both of them occur inside the nucleus. The lesson finished. Thank you.